Well, good afternoon. I'm glad that a lot of people could make it. Today we're going to be talking about the language of life and DNA, and we're specifically just going to focus on the matrilineal and patrilineal lines, the Y chromosome in the cell. You've got the nucleus, which has got 23 pairs of chromosomes, and you've got the mitochondria floating up all around in the cell. The test that a lot of us did gives us the DNA in the nucleus and the mitochondria. The mitochondria, because it's a pure female line, works like I just described. And because the Y chromosome is a pure male line, it works the same way I just described, except it's just male. The rest of the chromosomes in the Y chromosome is in the nucleus. The rest of the chromosomes that are in the nucleus give us a whole bunch of DNA information, and that describes our ancestry in much greater detail. So, for this talk, I'm just talking about the mitochondrial and the Y chromosome. The rest of the information is really interesting too, and that'll give us ancestry and a lot of other information. So before we start, I wanted to give you a general overview of what I'm going to cover. So what I'm going to do is I'm going to look a little bit about our ancient history to try to get a framework for what kinds of time periods we're talking about for the origins of these lineages, and then cover a little bit of basics of how we look at this DNA testing, why we can get information from it that give us these lineages, and then specifically cover mitochondrial DNA. And because there's so much overlap, there's so many things that are in common when you look at a single lineage that you can only get from one gendered parent, that when I get to the Y chromosomal DNA, a lot of the basic concepts will already be covered, so that'll be a lot shorter. And we'll have breakout sessions for both of those, and we can look at our own lineages. And if you don't have your information here, that's perfectly fine, because we've got plenty of things that you can do in discussion with others, including supplying you with a bogus lineage that you can just use for yourself that's randomly assigned. And then we'll cover a little bit about why this is interesting, at least why it's interesting to me. I mean, everyone can decide for themselves if it's interesting. And hopefully have just a little bit of more time for discussion. So, moving on, the first thing I wanted to do was give you a basic overview of our history from the Big Bang until now, and I'll mention when mitochondria come on the scene, when they first evolved. So our mitochondria are really a connection all the way back to that time. So I've got a real quick overview that covers in about maybe just a little over a minute our whole history, and I'll indicate when the mitochondria are evolving there. Life has had mitochondria for a long time. So a quick history. We're going to go back for a brief story going back to the early Earth. We go back here 2 to 3 billion years, and we're looking at the early Earth before there were animals, before there were plants. All that we had on Earth were single-celled organisms, or bacteria, and those filled the oceans and the shorelines. We also, in the atmosphere, only had nitrogen, methane, carbon dioxide, didn't have any oxygen. So let's look at what happened back then. Back then, bacteria are really good at evolving different chemical routes of doing different chemical reactions. We use those every day for our metabolism and a lot of other things in our bodies. And at that time, they had lots of food sources in the ocean because we still had all these organic molecules left over from before life started. So the bacteria would eat that. Well, those started to get used up, and after a while they started to run out of food. So they're trying to find chemical ways to get food energy. And what they found was there was a mutation at one point in a bacterium that allowed it to take the energy from sunlight and use it to crack apart the water molecule of H2O to take the H off and the O off and use the hydrogen in chemical reactions for food. We call those algae. And the algae that did this, the basic, and they ended up being with us today in plants as well, they started doing this to get a lot of their food. Of course, there was just one at first, but then, of course, it divided. You had 2 and 4 and 8 and 16, and pretty soon these things are all over. And the waste product is just discarded. It's just oxygen. They didn't need it, and it was a waste product. In fact, for these bacteria, because they were anaerobic bacteria, it was toxic waste. This toxic waste product, they just spewed into the atmosphere. So at this time, billions of years ago, we had a life form producing tons and tons of toxic waste and dumping it into the atmosphere. And the consequences were pretty significant. What happened right away was it started to kill off a lot of the other anaerobic bacteria. So bacteria were dying right and left because of this oxygen. And things even got worse because other bacteria and other single-celled organisms are also looking for ways to find food. And they found that instead of just picking up food that's lying around, they could actually start eating other organisms. So they started to eat other organisms, and so we had predation going on, and we still had these toxic oxygen clouds. Well, these here are bacteria, modern bacteria, the little red spots right here, and they're known as Rixetta. They're the closest relative living today to a certain kind of bacteria that lived back then that evolved a very interesting and very useful chemical ability. And that was to detoxify oxygen by using it in a metabolic system. Detoxifying oxygen just like our livers detoxify alcohol. And these little bacteria here started to detoxify the oxygen, and in that way they were okay and they were able to survive. However, it so happened that about that time they're still pretty small, and one of these larger organisms ate one of them. And instead of digesting it, it managed to survive in there. I'm sure that a lot of them were simply digested, but at least one wasn't. And the situation arose where it stayed inside this larger cell. It stayed inside this larger cell, and being there, it was protected from being eaten by other predatory organisms around there. And it started to mop up that oxygen in that cell, keeping that cell safe from the toxic oxygen. It entered a symbiotic relationship. So it gets eaten, we get this symbiotic relationship, and as the larger cell divides, it's got all these bacteria in it. They're dividing too, and so each half will have descendants of the smaller bacterium, and so pretty soon all the cells have that, which is a good thing because by now there's lots of oxygen in the atmosphere, and if you can't handle the oxygen, you're in a lot of trouble. So that story was figured out by Dr. Lynn Margulis around in the 70s. And she encountered an awful lot of resistance because this was a pretty radical idea to think that we had this symbiotic, relationship with these bacteria. And it was especially shocking because the descendants of those larger cells eventually clump together to form plants and clump together to form animals. And that means that all animals and plants have these bacteria in them, in their cells, taking care of the oxygen, and we call them mitochondria. If you look at the DNA between them and the Rixetta bacteria, it's clear where they came from. And there's a lot of other pieces of evidence that make it clear that this is really what happened. So another case where a woman had to really push to get her scientific idea accepted, one of the biggest ideas in biology. So that's where we get our mitochondria. And I find that story to be amazing to think that we've got in all of our trillions of cells, hundreds, often thousands of these bacteria. If you look at all the DNA in your body, the majority of it is bacterial DNA. Because even just looking at the mitochondria, there's, like I said, hundreds of thousands of these in every cell. In fact, I'm not the only one who found this to be an incredible idea. Someone else did, someone you guys know, have heard of, and that's George Lucas. George Lucas heard this idea, and he thought, wow, mitochondria, I wonder, giving us power to metabolize oxygen, being able to fuel our whole bodies in a symbiotic relationship in each of our cells. He thought, man, that's so incredible. That's even wilder than something you could just make up. This is a case where truth is stranger than fiction. So he said, you know, what if I made a whole fantasy world where that was the backbone of the story?
Master, what are midichlorians?
Midichlorians are a microscopic life form that resides within all living cells.
They live inside me.
Inside your cells, yes. And we are symbionts with them.
Symbionts?
Life forms living together for mutual advantage. Without the midichlorians, life could not exist, and we would have no knowledge of the Force. They continually speak to us, telling us the will of the Force.
The Force?
The Force is what gives the Jedi his power. It's an energy field created by all living things. It surrounds us and penetrates us. It binds the galaxy together.
So in the first part there, we saw the young Darth Vader's Anakin Skywalker asking about midichlorians because George Lucas had used the idea of mitochondria for his idea of calling them midichlorians that lived in the cells in his Star Wars world and made the Force happen. So the whole backbone, of course, to the Star Wars trilogy, or more than that now, is this idea of the Force, and he based that on these on mitochondria giving power in the form of midichlorians, is what he called them. By the way, they check Anakin Skywalker's, Darth Vader's midichlorian count. It's higher even than Master Yoda's. Mm-hmm. So here is a drawing of mitochondria. One of the many pieces of evidence that point to their bacterial origin is the fact that they contain their own DNA. As I mentioned before, that's where most of our DNA is. If a cell is going to envelop a bacteria, that bacteria starts with DNA, that DNA stays in it, and so all of us have DNA inside our mitochondria, and that's what we're focusing on for half of our attention today. So, to look at how we can test that DNA. This is our basic DNA. If you're not familiar with it, what we have is, you've got a chromosome. The chromosome is a different shape of bacteria, but at any rate, you start with the chromosome, and then you've got that spiraling double helix. The sides of the ladder are made up of phosphate molecules. The rungs are these nucleotide bases that we give the names of A, T, C, and G, depending on which molecule is making up that rung. So those rungs, if you look at them in order, you get a genetic code, and you get a series of letters like we have here. It goes G, C, C, T, A, G, something like that. So it doesn't seem to make a lot of sense when you just look at it that way, but scientists can decode some of it. There's still a lot of it that's a mystery. So if I looked at my DNA from my mitochondria, it might look like that top line. And then if I looked at someone else's, if I looked at yours, it might look like this next line. And of course, almost all of it's going to be the same. Better than 99.9% of our DNA, both in mitochondria and the autosomal stuff in the nucleus, is identical from person to person. We're just looking at the places where there's mutations that are different from person to person. So if we looked at this, we might say, okay, that's almost the same, but if we look really carefully, we might find a place where they differ. Here we've got GATA, and now we'll look at GTTA. And A is mutated into a T. Or did the T mutate into an A? We don't know which way this went, right? So we look at another one, and we say, okay, well, what's that one got? And it's got the T there. So we go, okay, well, we got two T's and an A. If these nucleotides were just in random changes, all over the population. You really couldn't make any sense of it, and we wouldn't be sitting here today. However, that's not the case at all. When you look at this stuff in detail and map out many, many sets of DNA, what you find is they form a pattern, a very definitive pattern. We get the first hint of it by looking at this set. Because if you look at that other mutational location, you've got TTCG, but if you look down below, we've got TTCC. So you'd say, okay, a G mutated into a C or a C mutated into a G. Well, which one is it? You look at this set and you realize that you only have these differences in cases where this one is a T. So maybe this one mutated from an A to a T first, and then later on you had this change. Maybe. It turns out that that kind of a simple logic sequence can take this DNA over dozens and dozens of mutations and build up a nested hierarchy. Now, a nested hierarchy is beyond the scope of the lecture here, but it basically means that whenever you have a certain feature, in the case of life, say something that spins webs, or in the case of these mutations, a certain mutation, you always have all the other ones that are designated by the circles that it's within. It's called a nested hierarchy. And the neat thing about a nested hierarchy is it forms automatically whenever you have descent with modification. That's why life is in a nested hierarchy. And when you see a nested hierarchy, it's a good sign that you might be talking about descent with modification. The neat thing about it is if you flip it on its side, it automatically makes a family tree of descent. Here we have a bunch of mutations that have been mapped out for bacteria, and again they make a nice family tree, where this lineage splits into these two, and then those split further, and you get the mutations like that, and you have a nice nested hierarchy, which I would encourage everyone to look into on their own time more. Okay, so that's how we can get these lineages, that's how we can get a family tree from these. And it's very different from just our regular family tree. It's different because it contains a lot less information. A lot less information for this reason. Everyone, I'm sure, is familiar with what we have here. And if you look at this here and say, okay, well, you know, where are the mitochondria? Well, we've got a mitochondrion there, mitochondrion singular. And in fact, like I said before, we have a whole bunch because we've got hundreds to thousands in a cell, certainly a big cell like an egg, and none in the sperm. There's a few in the tail, but they get discarded and left outside. So that means that after this egg is fertilized, the mitochondria are going to be from what was there before fertilization. They're going to be from the mom. The dad can't contribute any mitochondria. And so that means that when you look at a DNA sequence, you're looking at the DNA your mom had, in her mitochondria, in her mom, in her mom, and her mom, all the way back. With the only change being the few little mutations you might get after a time. And because there's a much smaller amount of total DNA in one mitochondria versus a nucleus, your mutational rate's really quite low. So what does that mean? Well, if we look at the number of ancestors we have, you can look at me, and I've got a mother and a father, and 4 grandparents, and 8 great-grandparents, and 16 great-grandparents. You can see these numbers get huge in an awful hurry. So you go back just a few hundred years, and you've got thousands of ancestors. And if you go back a few hundred years before that, you've got millions of ancestors. How many of them contributed mitochondria to me? Not very many. Just the mothers of the mothers of the mothers of the mothers. Just that one line. So if I go back a good ways, when I've got millions of ancestors, you can see that line I've drawn an awful thing here for a good reason. That's one person out of thousands or millions of people, depending on how far you go back. You go back maybe 800 years, 900 years, and you're talking about millions of people. If you take that whole pool of millions of people who were alive back then, who are your ancestors, and you reach in and pick one person, the mother of the mother of the mother of the mother, all the way back on the top of that line. Take that one person, take a look at their mitochondrial DNA, you've got an almost exact copy. In fact, if we're talking 800 years ago, it's almost certainly an exact copy. because the mutations don't happen that often. So you can see why this doesn't necessarily represent your overall ancestry, because you're talking about one person. If you're someone whose lineage whose lineage overall, your ancestry overall, is almost overwhelmingly one type, like for me, I look at my lineages, it's overwhelmingly European. If I pick one person out of the millions of ancestors I had 1,000 years ago, it's awfully likely that person's going to be a European. So it's not a surprise that it matches up. But because it's only one person, if by chance that person happens to be maybe the thin line of my Native American ancestry or something like that, then all my mitochondrial DNA would be like that. So it doesn't really represent your overall ancestry. That didn't happen with me. This is my diagram here. You can see this faint color here. It's just this little line of Native American on these two spots. And you can see they're close to being the top, but they're not quite the pure female line. One male in the line, and you don't have those mitochondria anymore. Because the mitochondrial DNA is only passed from the mother. So that means that over time, a lot of these lineages here, if you start off a long time ago with a whole bunch of different kinds of mitochondrial DNA, then you can see that over time some lines are going to stop just because you don't happen to get the mitochondria to the grandkids because you don't have a pure female line, or to the great-grandkids. And if you do, then it's got to just be lucky and have pure females again, or it's going to be lost. A good example is my family. I look at my family. My parents had 4 kids, 3 girls, and a boy. So you'd think, wow, that's pretty good odds to get lots of mitochondria, you know, several generations forward. Well, one of my sisters doesn't have any kids. Another one of my sisters only has no girls. Another sibling, of course, is me. I don't count at all because I'm a boy, so nothing's going anywhere there. And of that other sister, there's only 1 girl. So you can see it's very easy to lose these lines. And so over time, you can see that over time, one lineage will just happen to take over everything. But if this is many more generations, if this is millions of years, or even tens of thousands, you'll start to accumulate little mutations and you'll be able to tell these 2 lines apart, or maybe this line from this line. And then we're back to where we started, where we have different kinds. That's how it's going all the time. Now, I want to address the idea of looking at the most recent common ancestor that all of our mitochondria have. Our most recent mitochondrial ancestor here, this woman here in black. I want to address that for a few reasons. One thing is that there's always someone like that if you go far enough back for any group of people. For all of us here, the most recent mitochondrial ancestor could be figured out, and it's probably much more recent than the one for all of humans, because with humans you'll have greater diversity. And you could also do that for a larger group than that. Say take all the great apes, all humans, bonobos, and chimpanzees, and other apes, and the most recent mitochondrial ancestor will be farther back. That's very different from our most recent ancestor, because our most recent ancestor would include places where we can get to that ancestor by a mother's mother's father's father's mother mother father's line instead of just the pure female line. So that's much more recent. And part of the reason I wanted to mention that, you can always pick a most recent, you can always find the most recent, you can't find the actual person of course, but you can define the concept for a most recent mitochondrial ancestor, because that's true for any group of organisms. And if we talk about all humans, it's 1. If we talk about humans and apes, it's farther back, and that's much farther back than our autosomal one. Part of the reason I mention it is that that concept is one that you'll sometimes hear batted around, and I wanted to avoid using some of the more commonly used terms because they're terribly misleading, because they pre-program you with a whole bunch of wrong ideas about this most recent common ancestor and most recent mitochondrial common ancestor. And you get the same thing if you look at Y-chromosome, and that would be a male whose pattern would be similar, except then you're just looking at pure male lines. Okay, if we look at that, the neat thing is that when we look at where we find different mitochondria types, we find them mapped out in certain regions are a lot more common, and they happen to follow the same family tree that we would build if we had no idea of any of this, And we were just looking at where the mutations are built that nested hierarchy. This is yet another example of how cool it is in science when several different ways of trying to get information give you the same answer or a compatible answer. This is how the lineages broke out, and based on the mutation rates and the known migrations from archaeology, again, they agree with each other, you get these times for when these splits happen. About 170, 130 years, 1,000 years ago, we had an ancestral group in Africa, and that group split out, and you had mutations along two different lines, giving us the N and the N groups, and all the way to where we are now. Okay, haplo, what? Let me define some terms for you. Haplotype. Let's start there. Haplotype. A haplotype is just the name we give a certain set of mutations, a certain kind of DNA, mitochondrial DNA. So if you take a mitochondrion and analyze its DNA and say, oh, this is this one here that I also tested over there, and I also tested over there, and found it there too. So we should call it something, right? We're going to call it, in my case, K2C1. So if you have these mutations, you're a K2C1. That's the haplotype. However, there are, because there's been so many mutations in such a big population, there are many, many, many of those haplotypes. It's a lot easier if you can say, okay, I'm going to talk about, you know, going farther back up the family tree. If I can talk about this branch of the family tree, what can I use to talk about that branch of the family tree? We can call that a haplogroup. That's a larger grouping of haplotypes. So that might include K1C2, K2B1A, and all these other ones that are just names we give certain sets of mutations on the family tree. Okay, some of the more common mitochondrial haplogroups. We've got Helena and Jasmine. These are names given popularly to these groups that are just numbered with letters because, you know, it's a lot easier to remember and think of a name than a letter. So that's why they put those names on there. And the names have been picked so that they start with the letter. So otherwise it would be pretty confusing, right? So some of the more interesting famous people that belong to some of these haplogroups. By the way, I am from the haplogroup K, so my clan mother would be Katrine. But, of course, all of my kids aren't going to be that, right? All of my kids are in the clan Velda.
Which is Heather's.
Yeah.
It's going to be the same as their mother.
It's going to be the same as the mother, right? Same as the mother. Here's that family tree. And again, what these little numbers are on here are specific mutations. They designate a place in the code where you can find a difference, a C to a G or something like that. And so, you know, when they look at this, they say, okay, if it has this mutation and this one, but not these over here, then it must be down this family tree line or something like that. So any questions before we go any farther? So we're going to break into groups pretty soon. Any questions or thoughts about the mitochondrial DNA? A lot of us know our mitochondrial haplogroup, but for everyone we'll be able to do this. Haplotype even. Yeah, did you?
I'm familiar with the haplogroup one. I had that done like 4 years ago. But I'm curious about the other one that you're mentioning that you said several people have done. Which test was that?
Oh, a lot of people did the 23andMe test that gives both the mitochondrial haplogroup, the Y haplogroup, and your autosomal DNA, which is the rest of the DNA.
23andMe.com?
Yes, 23andMe.com. Yep. So, yeah, your mitochondrial DNA doesn't tell you a lot about your ancestry because it's not necessarily representative. There's a good chance that it'll line up just... Okay, how many people know what these are? Okay, a few people. Good, good. That one on the left kinda, whoops, sorry, kinda looks like an X. And the one on the right doesn't really look much like a Y, but that's what they call it. It's got our Y chromosome for us men have a lot less DNA. The X chromosome has a lot of DNA on it that's absolutely essential to survive. You couldn't live without having at least one X chromosome. However, the Y is not like that at all. You can live quite well without a Y chromosome, as half of the world's population shows us. Okay, we saw before how the human population moved, and it would make a lot of sense if we see the same thing when we look at Y populations, if we see the same kind of migration. That one looks somewhat like an X, that one looks somewhat like a Y.
Looks nothing like the one.
Maybe. That's why they call it that. Okay, so let's take a quick look at this history again. And this time I'll show you when the Y comes on the scene.
Here we go.
Okay, here we go. Nucleosynthesis, formation of elements. Okay, here's mitochondria. Okay, still now we've got sexual reproduction, but still no Y chromosome. Still no Y chromosome. Still no Y chromosome. Now we've got no Y chromosome. So we've had that Y chromosome for a lot less time. Still a long time though. If you look back, our Y chromosome dates back to the time the dinosaurs were around. That's when mammals developed the sex determination system of XY. There's a lot of other systems in the animal world that are used. It's surprising that you think, oh, Y chromosome male, but no, that only applies to mammals. If you look at other sex determination systems among the only dinosaurs we still have around today, the birds, it's a completely different system. And a lot of animals have it based on environmental cues or things like that that aren't even chromosome based at all. Like for a crocodile, it depends on the temperature that the eggs sit at while they're incubating. So we can date our Y chromosome only back to the dinosaur time, when our ancestors looked very different from how we look today. Actually, we've got a representative of something like what our ancestors looked like in that picture. Do you see him or her? I'll help you out a little bit. Look at that. A lot smaller back there. Okay, we're back to the sperm and the egg, right? So, where are the Y chromosomes? Y chromosomes are in sperm, right? Well, yes, half the time. Only half the time. Half the sperm don't have a Y chromosome. They have an X chromosome. And of course, the X chromosome is what's present in the egg, not the Y chromosome. So, just as we saw with the mitochondria, this is only going to be passed along a single gender line. It's going to go, in this case, male, 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 male. And so, all the stuff we had before applies, a lot of stuff that before applies, in the sense that our most recent common male, male, male line ancestor is going to be much farther back than our most common ancestor in any case. So again, you can't really look at ancestry much with your Y chromosome DNA data. Or if you're female, with the data you might get from a brother or father who can give you that same male, male, male lineage. Because, just like we saw before, you go back just a little ways and you've got millions of ancestors. And of course, you're only going to count the male, male, male line right here. And again, have that real thin line when you get back a long ways here. Again, we're going to be picking up one ancestor out of millions when we go back a few thousand years. And again, just like the mitochondria, the amount of genetic information on there is really pretty small compared to, say, the autosomal DNA. So that means you're not terribly likely to get a mutation at any time. And you might go many, many, many generations before you have a change that shows up that people notice. And that's very different from our autosomal DNA, where we have 3 billion base pairs. And the odds are that all of us here have some mutations that our parents didn't have. Okay, this is what we're looking for, right? Do the patterns of migrations and the times match what we see with the mitochondria? And it does. Again, we get the case where we build up a nested hierarchy based on the genetic test, just like we did before, and that builds a family tree. And you take that family tree and you look at the times that are designated by the molecular clock, and you find the same patterns of migration shown by where we find that Y chromosome today, where it's most common. And it's really, I find it really powerful, although not surprising, that we see the same pattern of migration we saw before, with approximately the same times. Here is that family tree made up using the Y chromosome data. And I'm in the R haplogroup, so I'm down here. We use the same terminology, haplotype, haplogroup, when we're talking about Y chromosomal DNA, because we get the same kind of dynamics happening again. Here's that family tree, again, put vertically, just like with the mitochondria, so that we can take a look at it. And we see, again, that the times we're talking about are in many thousands of years, often dozens of thousands of years before present, just because we're talking about a smaller piece of DNA than autosomal DNA, so your mutations are more rare.
John, should there be lines that go down from the E and G to modern times, or do those actually die out at that time?
That's a great question. Yes, there should be lines that go down to modern times, because all of those are extant as far as I'm aware. Extant meaning we still have people around today that have those Y chromosomes. Interestingly,
What about T?
T, yes, I'm pretty sure there's still T's. Check on that one.
Does it have other mutations?
Right. Oh, are there subgroups to T? I think so. This is terribly simplified because a lot of us, Joseph, you remember yours, right? It was, it had about 8 letters in it. It was something like R1, B1, B1, A1, A2, A1, F star, or something like that.
That's like mine.
That's like yours, yeah. And that's like mine too. Mine ends D asterisk. But anyway, put the asterisk on there because they find, you know, everyone has that same sequence of mutations to give it that name. And then they find a subgroup that has that and another mutation. What are they going to call them? Well, we'll put an asterisk on it. And it's, I can imagine how hard it is to, you know, start grouping these into a family tree and come up with names for them. A lot of the R chromosome, Y chromosome data haplogroup is very, very confusing. One thing that I find is useful for anyone who's checking either themselves or a father or a brother or something like that, for the H haplogroup, for instance, in mitochondria, there's a ton of information. With the Y, a lot of these are small enough subdivisions that there's not a lot of information out there. So a lot of times what I find is useful is when you get the result, just type the whole string into Google and hit search. And anyone who's typing that search is talking about, you know, on some page or something, is talking about that haplogroup, so then it's easier to find interesting stuff. A friend of mine, Michael Dow, we pulled up his data. I was going through it with him, and he looked at it and said, well, you know, what's that mean? And I said, I don't know. It's, you know, some long string of letters and numbers. So we took that thing and popped it into Google and hit search, and it turns out that it's the Y haplogroup from the High King of Ireland, who lived over 1,000 years ago and had a lot of male descendants. And so that means that my friends not only descended from that High King, I mean, after all, most of us are certainly any Italians, Irish, but not only descended from him, but descended along a pure male line all the way to him. Wow, interesting. And you can find the same thing, you know, sometimes things like that will pop up with yours. On a side note, it's worth noting that because of the sheer number of ancestors we have, you remember I showed that you go back 1,000 years, and we've got millions of ancestors, right? Pretty much all of Europe. And so that means that for people with European descent, all of us are descended from kings anyway. It's just not along a pure male line. So all of us are descendants of kings and you know, of course, tons of peasants, but all kinds of, almost any group. You can certainly pharaohs, popes, stuff like that.
You know, there is a famous king, the most famous king of Morocco, Mulay Ismail, and he had thousands of children, hundreds of women, but he killed most of his girls. He didn't value girls, so I mean, he really did a disservice to genetic research by killing girls and leaving only the
Yeah. Well, another good example is Genghis Khan. We've identified the Y haplogroup of Genghis Khan, and it's really pretty common.
There's like half of the world.
Not like half the world, but it's getting there. There's a lot of people descended from Genghis Khan on a pure male line, and an awful lot of people descended from him anyway. So, okay, so a lot of that information is the same. I should point out that the most recent Y chromosome common ancestor for all the male lines, all the men, was thought to be for humans back certain ways until maybe 100 to 200,000 years ago, until just last year they came across a Y haplogroup that didn't fit the chart at all. Anyone. And it had mutations that showed that it was a whole separate branch. So they had to estimate farther back. And that shows that that line was still around there. And so that, and in the same way, as more people get tested, it's more likely that we'll find odd mitochondrial or Y chromosome haplogroups that push the common ancestor farther back along the pure lines. And as I said before, that's not the same as our most recent common ancestor. For humans, all of us have a most recent common ancestor back on the order of thousands of years ago. Nothing like a lot of the numbers we have here, because that's on a line that's not a pure male or a pure female. So any other questions before we go to the next activity and breakout group? Okay. This is the overall migration pattern, and these are the dates by thousands of years when these different splits happen by the different colors. So you can look on here and say, oh, I've got haplogroup R1A, and someone else has haplogroup R1B, and if you look there and say, when did they split, that's this color right here, and it looks like about 10 to 25,000 years ago, which, in terms of haplogroups, is a lot. Interesting. Now we've looked at mitochondrial and Y, and we are going to move on just for a brief time. I asked the question, why? Why do we even do this? Why are we all here in this room? Why is it interesting to look at our mitochondrial and Y DNA? Well, probably not for medical reasons. Because like I said, it's a small amount of DNA, and there are problems with medical problems that can happen with problems with especially the mitochondria, but they're really quite rare because there's not a lot of DNA there to mess up compared to your autosomal DNA. What about ancestry? As I mentioned before, it's not really representative. It doesn't give you a lot of detail, and the piece of information it does give you may or may not be relevant. It just tells you in the broadest terms what you might be overall. And of course, it's soon lost. If you think about it a little bit, you realize that both your mitochondrial and your Y chromosome lineages are probably irrelevant to your descendants. If you have descendants, or if you think about, you know, a brother's descendants or an aunt's descendants or something like that, in any case, your kids are probably, you know, your male kids will have your Y, but then if they have a bunch of daughters and then, you know, after that, that Y doesn't continue anymore on your line, and the same thing goes when you look at it reversely from the female side. So it really doesn't tell you much about your descendants. Very soon, if you have some descendants, very soon they have the same population frequencies as wherever they are over the span of, you know, hundreds of thousands of years.
Fierce.
So why? So why do we look at this? Why, which is a short time, right, when you compare it to longer geological times. So why do we look at this? Why do I care? Well, that's, of course, for everyone to decide for themselves. But for me, I find these lineages to be really interesting, too, in addition to the autosomal DNA. For one thing, they can connect you to times way farther back than your autosomal DNA because they're preserved for so much longer without a lot of changes. This is Aussie Oxy was found in the Swiss Alps when about over 5,000 years ago, he was killed by getting shot in the back by an arrow after killing several other people himself. And was only, and this is, Amy, that's a great question. How did they know that? Because they did DNA tests on the flint dagger that he had, that had blood from a human on it, and some of his arrows had blood from 3 other different humans
So he
that
was
he
there.
had. And it's easy to tell from, right, when we have the body, it's easy to tell if
He got shot in the back.
he got shot in the back with an arrow. And a lot of it, this guy is the same haplogroup K as me.
I
So
can
it's not a
tell.
rifle? It's not a turtle?
No, it's not a right there.
It's not a rifle. That's a squiver of arrows. That's a squiver of arrows. Yeah, and if you look, he's got a digital watch too.
Yeah.
But yeah, no.
Aside from that.
Not exact, right. But, you know, I look at all the stuff he had and how he had to live, and that's a connection.
He's pretty sophisticated.
He's geared up for the snow. Up walking in the Alps long before anyone called them the Alps. And that's a connection. We can really see some deep history here when we look at our mitochondrial and Y haplogroups. And that's not, I'm not the only one. There was a skeleton found in a cave, back in a cave in England, known as Cheddar Man. And they did DNA tests of people and found someone living right there, a kindergarten teacher, had the same mitochondrial haplogroup. So it's a connection to a lot deeper time past than you might get from your autosomal DNA, just because a lot of that gives you information more recently. But of course that information had to come from farther back too. I don't want to make it sound like that just popped out of nowhere. But the point with the mitochondrial and Y is that it only gives you information from way farther back, because people carry it without much change for a long period of time often. And I find that to be really interesting to look at. It can connect you farther back than that. I look at the story of how we got our mitochondria, how all life that has mitochondria got it. We got it from these bacteria inventing the way to detoxify oxygen 2 to 3 billion years ago. Wow, that's really neat. And we've got it over and over and over, thousands of times, hundreds of times in many of our cells all through our whole body. What a neat story. It also, the Y-chromosome also connects us to times 100 million years ago or so when that sex system was first developed. Wow, that's really neat too. To imagine what it was like back then for our ancestors to survive by cowering in fear from the roar and noise of these terrible lizards. And we can't forget that for both the mitochondria and the Y, we have incredible stories of the scientists who figured this stuff out. It's not simple. It's hard enough for us to just look at this and try to drink in all the information now. To imagine having to discover it yourself from scratch, that's hard. I look at these courageous stories and I think, man, people really put in a lot of work to figure this out. And I get to benefit from it, and I didn't even have to do it. Wow. I find it to be interesting to think of that, really intriguing. So if you also get that kind of a charge from this information, then I hope this session was interesting and fun for you. That's the talk. I will have one short breakout session. Let's have one short breakout session, but before we do, I wanted to point out something else. A lot of you know me. I'm John Cleveland Toast. Cleveland helps honor my matrilineal line. Actually, not mine, but that's, for our family. That honors the matrilineal line for our kids, because that's Heather's mom's maiden name. We couldn't go all the way back, of course, because there are names. But that has a connection to our mitochondria. And of course, for most of us, our last name is passed along a pure male line. And that has a connection to our big and strong Y-chromosome.
Yeah. Looks like a bunny.
That's funny. So, let's have a couple questions and thoughts, and then just a short time we can talk about anything from this whole class before I finish up. Any other questions, thoughts? Yeah, Joseph.
Could you say a little bit more about autosomal DNA? I mean, because that's most of our DNA.
Yes.
He doesn't say much about it.
That's a great point.
Well, you
Joseph
probably-
asked about the autosomal DNA. I'm going to have a whole session on that.
Oh, thank God.
And I will cover it in detail because that's a huge topic. The autosomal DNA is incredible. It's really neat to look at what that autosomal DNA does for us. It's really interesting to look at that for recent ancestry and more deep time ancestry because it gives us information on all of it. After all this DNA, whether we're talking about autosomal or the sex-linked lines, goes back a long, long time. Looking at all the connections, all the interesting things that this DNA tells us, and it means a lot to me. I hope it means a lot to you too. And so with that, I'll finish up and say thank you. I want to put in a short plug. Remember, everyone, this was part 1. We've got a full session on the autosomal DNA. I'm going to have a regular Sunday service where I'll give a talk and presentation about what it means to me on a meaning and purpose level, and then afterwards have a session like this to go through a lot of the details that will be structured similarly to this one. That's July 13th this year, when we can take a look at our autosomal DNA. Here's some compositions from people. This one over here is me, and
Which one are you, John?
what am I? Well, it's depending on how you split it up.
Right.
Almost all European. But if you look here, the way that they split up their reference populations, I've got almost all North European, a good chunk of Mediterranean European, a tiny sliver of Native American, and the other ones are smaller groups that include some drift over from Asia and other parts of Europe. And this one here is by chromosome. This isn't me. This is someone else, a friend of mine, whose DNA is mapped out by chromosome. We can do, all of us, a lot of us know we can do this for ours too. I can do it for mine. It's almost all blue, of course, because that's European. But this shows what chunks of ancestry are on what chromosomes. So you might have a chunk of European on chromosome 5 or a chunk of Asian on chromosome 11 or something like that. And on the 13th, July 13th, we'll get into this in much greater detail. Yeah.
I just want to clarify, the biggest difference between autosomal DNA and the Y and the mitochondrial is that the only changes we get from mitochondrial and the Y is the mutations over time.
Right.
With autosomal, you can get it from either parent, and that means there's also crossover happening. So these things recombine in different ways, so they have a lot more complexity.
Right. Yes, that's right. So with that, thanks for coming.
Thanks, John.